viewers.
So thank you very much, Dr. Vivek Asati ji and Dr. Preeti ji. So first of all, I welcome today's expert guest, Dr. Reem Arafa at ISF College of Pharmacy in India. So I feel that this is the one of the proud movement of this institute. Such types of the experts, they are attached with this institute and they have accepted our invitation to deliver a talk on a very specific topic, a journey in the discovery of the anti-cancer drug. So again, I welcome from core of my heart Professor Rimaji, and I am thankful to our core committee for provide the opportunity to interact with you. So without taking time, I request to Dr. Vivek Asati ji, please proceed.
Okay, sir. Thank you, sir. So I request to Professor Arafa kindly deliver expert talk on a journey in the drug discovery of anti-cancer drugs. So, ma'am, now you can start.
Thank you so much, dear professors, for your kind invitation, and it's a pleasure to be with you today. Um, I was really impressed to receive your invitation, and it gave me a lot of pleasure to get to know eminent professors like yourselves, and across the distance. And Egypt and India are geographically far apart. Yet science brings people all together, and our passion for science collects us to share our knowledge and participate in common talks regarding what we do. Um, Please allow me to share with you a couple of steps about the journey of drug discovery of anti-cancer drugs that I started long time ago, 1997 and onwards. And thank you for the kind introduction. It was very generous of you. Um, I'll brief the work out in two phases. The first approach that we use is conventional drug discovery, and I'll share with you today the discovery of new selective COX-2 and EGFR dual inhibitory oxidazoles as anti-cancer agents. Later on, I'll be sharing with you a more updated approach of the drug discovery process for anti-cancer agents. The first approach, I'll talk briefly about the introduction, research objectives, experimental work, biological screening, and the in silico study. And we'll conclude this first part with a conclusion and recommendation. As you all know, cancer is a set of diseases, and it affects any part of the body, any organ of the body, and that's why it was called disease. It's not only a disease. And calculations and statistics say that number of deaths in cancer are in all types of cancer and in both sexes at all ages is really hitting hard all parts of the globe. Asia, Oceania, Latin America and Caribbean, North America, Africa, Europe. People all around the globe are being affected by cancer. When we say that, we ask ourselves what are the most prominent types of cancer so that we try to fetch for cure for that type of cancer. We found that renal cancer is one of the most aggressive types of cancer, and when they hit hard. The diagnosis is a little bit late, and it is life-threatening. As you can see here, according to the World Health Organization, in 2017, kidney cancer came third after, of course, breast, prostate cancer, and colorectal cancer. Yet the bad side of it is, as I mentioned earlier, that when it hits at a lethal stage, and usually if not diagnosed early, it can lead to metastasis and to death. I think there is something wrong with the slides. They keep coming back to the same thing. So allow me to a little bit escape and then come back. According to the Global Cancer Observatory, statistics showed that incidence and mortality of cancer are higher in males more than females. According to the American Cancer Society in 2019, it showed again the same pattern where incidence and mortality were higher in males than in females. And that calls for an intervention. And there are too many cancer treatment modalities, not only for renal cancer, but for other types of cancer as well. There is the hormonal therapy, surgery, bone marrow, transplantation in case of blood types of cancers, chemotherapy, targeted chemotherapy, radiation therapy, immunotherapy. However, the focus of our lab was always on finding targeted therapy, which is a more updated and safer option than chemotherapy, which has a lot of side effects. So in that case, we search for what are the biological targets, those kinds of proteins or enzymes, receptors, or biological molecules that we can target to stop cancer progression, and in effect, if we can, as well kill the cancer. So we found that there is a strong correlation between cancer and COX-2. COX-2 is that enzyme that is in general related to inflammation, and it has been COX-2 inhibitors have been used for long for treatment of inflammatory diseases like bowel inflammation, wound inflammation, tooth inflammation, and stuff like that. However, there is a strong correlation, as I mentioned, between COX-2 and cancer, which was recently proven. COX-2, it has the ability to convert, it's an enzyme, it has the ability to convert arachidonic acid into prostaglandins, and prostaglandins, they increase the angiogenesis, proliferation, anti-tumor immunity is reduced, and apoptosis is reduced in cases of cancer. So if we find molecules like non-steroidal anti-inflammatory drugs that can work to inhibit that enzyme, they can block the formation of prostaglandins and eventually can reverse all the things that are associated with cancer, which will lead eventually to cancer cell death. On the other hand, we found that there is another enzyme really important. It's actually a receptor and enzyme at the same time. It's called EGFR, short for epidermal growth factor receptor. And it has a big role that is associated with cancer. As I said, it's a receptor, and it's embedded in the plasma membrane of the cell. It has an extracellular part that binds to the epidermal growth factor. And once bound at the extracellular part, this activates an enzymatic activity at the intracellular part of the EGFR, and this calls for what we call a signaling cascade. EGFR belongs to a family of enzymes known as kinases, and these kinases are known to phosphorylate a series of targets, biological targets, and the phosphorylation of which leads to switching off switching on of their activity and a signaling cascade. At the end of that signaling transmission, cells will eventually be able to proliferate, and cancer cells will be invasive. This will cause metastasis and secondary types of cancer, and it will halt apoptosis. Apoptosis is the programmed cell death, so it will prevent cells from undergoing death. Thus, if we inhibit this epidermal growth factor receptor from doing this big job, we will reduce the proliferation of the cancer cells. We will induce apoptosis of these cells, and of course, the invasiveness of this cancer cell will be reduced. So the idea we had in mind is to find a new scaffold that can act as a privileged motif with the ability to bind to both COX-2 and EGFR at the same time. So we went into the literature and made some search. We found that the COX-2 inhibitors, they share a common 5-member heterocyclic ring, and that's a small-sized ring, and it can be easily decorated around. So, for example, the drug that is used in the market, celecoxib, it has the imidazole, and rofecoxib has a furan ring, and valdecoxib, it has the isoxazole ring. This is an investigational drug that has a thiophene, so we thought, why not explore that privileged motif, and we do some decorations on it. We wanted to make sure that also they have the ability, the oxidizole, the ring that we are targeting to use, which is in that case 1,3,4-oxadiazole. We have expertise in synthesizing at the lab, would be able to have an anti-inflammatory and an anti-cancer effect. So we fetched further in the literature, and we found that, oh yes, indeed, the oxadiazole has an anti-cancer effect and anti-inflammatory effect. And of course, that hits the target that we want in that case. So the design strategy was based on a drug that we found in the market. It is called Decoten-10, and this drug has an oxidizole ring, and it acts as a strong anti-cancer agent. And we thought that the oxidizole in our case has a medicinal chemistry advantage of ease of synthetic approach to it, and it has a good medicinal value. It has the ability to do many ligand interactions. It's kind of flexible in the synthesis, and the polarity is nice. The pharmacokinetics, I mean absorption, distribution, metabolism, and elimination associated with having this moiety in a drug is promising. In vivo metabolic profiling of that part of a molecule is really also supportive of our hypothesis. So we found that the EGFR inhibitors that are published, they have an oxidizole ring, and they are substituted at the 2 and 5 positions, and they have an IC50 of 0.081 millimolar. And we wanted to improve about around this number. I'll show you our data later. But again, the binding interaction of this molecule with EGFR showed that the oxadiazole, the 2 nitrogens, they have additional supportive importance, which is a binding to the receptor active site amino acids, along, of course, the patterns of substitution on the 2 and 5 position. In that case, we came up with a design strategy for our target, 134 oxadiazoles, and we wanted to decorate at the 2 and 5 positions with hetero-aryl or aryl 6-membered ring at one side. In that case, we used the phenyl, and it's either phenyl or chlorophenyl, and pyridine, either the 3 or the 4 pyridyl substitutions. On the other side, we had a linker, spacer, and we had a diversity of linkers being tried: rigid cyclic, rigid acyclic, flexible short, flexible long types of linkers. And eventually we had at the other terminal different chemical scaffolds that were tested. Phenyl with the different substitutions at that end, substituting it with the chloro, bromo, nitro, or hydroxyl, or non-aromatic kinds of chemical scaffolds like cyanomethyl or ethoxy groups. So in that case, we initiated a research that focuses on new hybrid molecules designed based on the reported EGFR and COX-2 inhibitors, and having, of course, the 1,3,4-oxadizole moiety as a privileged scaffold in that case, with the substitutions at the carbon 2 and carbon 5 positions of the oxadizole skeleton. After that, when we synthesized and proved all the validity of the structures that we had in mind, we tested them against COX enzyme, and we tested them against EGFR, and we tested them against the target cancer cell lines in that case, which are the renal cell carcinoma. And eventually we concluded with a lot of focus on the in silico study on both enzymes COX-2 and EGFR, and we did a study on the molecular dynamics as well. To test about the druggability or the ability of our molecules to be a good drug-like compounds, we evaluated their in silico ADME profiles as well. So for the chemical synthesis part, we had 3 schemes. We started with this compound here, an oxidizole with a mercaptan group at one position, and with a 4-pyridyl substituent at the other, at one terminal. And we converted the mercaptan group into a hydrorazinyl group, and then we reacted them with isocyanates, or compound 1 was directly reacted with a spacer, and we either carried this hydrazine and had Schiff's bases by reaction with aldehydes, or the hydrazine that is directly attached to the oxidizole and reacted it with aldehydes, and we ended up with 2 series of Schiff's bases, one with a small linker, which is the series 3A to D, and one with a longer spacer or linker, the series 6A and B. And eventually we wanted to test the hypothesis of introducing another oxidizole ring. So we got those compounds, derivatives 5, and cyclized them by reaction of substituted aromatic acids and in the presence of phosphorus oxychloride. And we ended up with having derivatives 7A to C. All these compounds were considered final compounds and were tested biologically for their COX inhibitory activity. Of course, and as the usual trend in medicinal chemistry, whenever we have new compounds that are not reported in the literature before, we need to validate them structurally by structural elucidation. So as an example, and I will not go through all the spectra, yet I want to share one example, which is compound 3D. For example, we go for, of course, IR. I know that this is the usual trend. Proton NMR, deuterated proton NMR to make sure that the NH peak is exchangeable, deuterated and exchangeable, and the carbon 13 NMR, and this goes for all the compounds that we synthesized. We went through and had another series of compounds that were synthesized through scheme 2, which is synthesis of 2 substituted 5,3-pyridyl 1,3,4 oxidizoles. In that case, we started off with compound number 8, which has amino at one side of the oxidizole ring and a pyridyl, but with the 3 substitution pattern, nitrogen at the 3 position in that case. And this was our starting molecule. We had a few Schiff's bases, couple of Schiff's bases from it. We had one compound with a short terminal that is not aromatic in nature, a slightly longer terminal that has a cyano group, and the cyano group would add a little to the pharmacokinetics of the molecule, and eventually another molecule that has a urea spacer and a phenyl terminal ring. As you can see, we were trying to test the diversity of the characteristics of substitutions and the linkers at one side, so that we can end up with a conclusion about the structure-activity relationship, which I'll come to later. In that same project, we also synthesized, as per scheme 3, 2 substituted 5-phenyl or 4-chlorophenyl 1,3,4 oxidiazoles, starting with the key intermediate 13, A and B, where A has hydrogen on the phenyl and B has a parachloro in the phenyl. And again, we exploited the amine functional group and had urea derivatives, same wise Schiff's bases, short linkers, non-aromatic, aliphatic, and also more flexible short linkers that are aliphatic in nature. So we ended up with compounds 17, 16, 15, and 14. Okay, so moving forward, we got all those compounds and started going to the COX inhibitory activity and testing the cytotoxicity and testing the EGFR inhibitory activity. And that's what I am going to discuss with you in the next few slides. All compounds were evaluated against both COX-1 and COX-2 because there are two isozymes or isomeric forms of the enzyme COX-1 and COX-2. You want to be selectively binding or inhibiting COX-2 because COX-1 inhibition will lead to some side effects that we don't want our compounds to do. So here is a table that is showing the tested compounds, all of them. And the COX-1 and COX-2 IC50s, they came in a very nice range, the micromolar range, or actually, to say the truth, in the sub-micromolar range, which is in the nanomolar. So all compounds are really potent. However, some of them were highly potent, eliciting a 0.053 micromolar activity. This is compound 7C, compound 14A, and compound 18B. We had this in comparison to the very well-known COX-2 inhibitor celecoxib. We also compared all our compounds with diclofenac sodium, which is a COX-1 inhibitor, and with indomethacin, which is a COX-1, COX-2 dual inhibitor. And we tested the selectivity index. It's something that is only calculable. It's not experimental. We divided the value of the inhibitory IC50 of COX-1 and COX-2, and we ended up with these numbers. These numbers, usually, the lower the number, the better. We can say that we have, the higher the number, the better we say that we can have selectivity. I mean, in that case, they have high IC50 towards COX-1 and low against COX-2. So the dose that will inhibit COX-2 will be much, much less than that will inhibit COX-1 in that case. As we can see an example here. The IC50 of 7C, for example, is 4.7 micromolar for COX-1 and 0.053 for COX-2, and thus the selectivity index is 78. From that, we wanted to find some SAR findings, and SAR stands for Structure Activity Relationship Fund. So what in the structure will have the compounds behave better towards COX-2? It came up that those are the three series that we had. But for example, when we have this scaffold constant and the spacer constant, we found that the nitro is more potent, gives more potent derivatives than the bromo, than the chloro, and than the hydrogen compounds. So electron withdrawing hydrophilic groups would give a better activity in that case. Same happened when we had a longer spacer, and the nitro was better than the chloro. And when we had an aromatic spacer, the oxadiazole, another oxadiazole ring, we found that the bromo is, again, more potent than the chloro as an inhibitor of the COX-2 enzyme. For the series of the 5,3-pyridyl 1,3,4 oxadiazoles, we found that lower COX inhibitory activity was elicited. So apparently an aromatic ring on this side, on the other side of the oxadiazole, I mean the 2 position, will enhance the COX inhibitory activity. For the 5-phenyl or 4-chlorophenyl 1,3,4 oxadiazoles, we found again that the aromatic ring with a nitro imposes good activity. A little bit difference here when we had the phenyl with a parachloro substituent on that side of the ring, which is the 5 side of the oxadiazole. Then the chloro was a better performance than the nitro, than the hydrogen on the 2 phenyl at the 2 position of the oxadiazole ring. in general, if we compare the substitutions and at the 5 terminal, we find that the phenyl, the pyridyl are more or less the same. However, the parachlorophenyl was a better inhibitor of the oxidizole COX-2 effect. So we went ahead and selected 9 of our compounds, structurally diverse from various, most potent from the various series, and we tested them against the renal cancer cell line that is known as UO31, using an assay that is called MTT cytotoxicity assay, and using doxorubicin as a reference drug. And here are the data that we had in comparison with doxorubicin, which is a positive control in that case, and it has a non-activity, anti-cancer activity. Our compounds here gave us a very nice information and very promising data, especially top 3 of them. Compound 3D gave us IC50 of 8.6 nanomolar. Compound 9 gave us 5.7 nanomolar. Compound 12A gave us 13.5 nanomolar IC50 in comparison to that of doxorubicin, which is 7.45 in that case. So we have 2 compounds that are one is a less more potent and one's almost equipotent if we calculate the SD values. And here is another promising compound. We went ahead and explored the structure activity relationship in relation to the anti-cancer effect on UO31. And we found that it came out that we have 5 of our compounds that were the most potent, and among them, the structural insights that we got is that smaller linkers, shorter linkers, either urea or CH double bond N linker, or even here, those coming from the Schiff's basis, the amine functional groups or the urea functional groups are better than the lengthier functional linkers or the more rigid oxidizole linker. So a little bit of lengthening or rigidity in that case are not in favor of the anti-cancer activity. In terms of difference on substitution at the 5 position, we found that the 3 pyridyls were better than the phenyl and the parachlorophenyl. In terms of the EGFR inhibitory activity, we went ahead and selected top 3 compounds from the cytotoxicity assay, which are 3D, 9, and 12A, and went ahead and tested them against the second target in that case, which is epidermal growth factor receptor. And here are the data for the compounds. The IC50s are again in the micromolar range, or actually in the sub-micromolar range for 2 of them, 0.4, 0.2, and 1.1 for compounds 3D, 9, and 12A respectively. So this is in line with the initial postulation that those compounds can inhibit both COX-2 and EGFR and have an anti-cancer effect or a cytotoxic effect on the cancer cell lines. They were compared to the very well-known compound EGFR inhibitor, erlotinib, and we found that 2 compounds, one was more potent and another was equipotent with erlotinib as a positive control in that case. So with that in mind and this information in hand, we went ahead and did some docking. And the docking here is to try to make us comprehend how these compounds bind to their target. If you recall, I had an initial hypothesis that they would bind through the oxidizole ring. It will play an initial important role in the binding, in addition to those substituents at the 2 and the 5 position of the oxidizole. Later, we did molecular dynamic simulation, which is really a second phase that we do after the docking, and the importance of this is it simulates the molecular motions that happen at the body in the presence of water, and it tests a longer period of time, and it tests if the compounds are forming stable complexes or they will break down under the motion and competitiveness of the water molecules with the active site and the ligand at the active site of the receptor. So we went ahead and did molecular docking with COX-2, and we selected the most potent compound in the COX-2 assay, which is 3D in that case, and we utilized the X-ray crystal structure that we obtained from the protein data bank. Its code is 3LN1. And the molecular docking studies showed that our compound that bind nicely to the active site in a similar fashion to that of the co-crystallized ligand. The co-crystallized ligand is the yellow one, and our docked compound is here that has carbons in the cyan color. And as you can see, we found that the nitrogen of the oxidizole indeed has an interaction with 3,5,16, and the side chains started to form hydrogen bonds. Here is a hydrogen bond with the valine 3,5 of the active site of COX-2. The nitro group of our compound occupies the same region of the sulfonamide of the of silicoxib. You are comparing here with silicoxib, and forms interactions with arginine 499. And as I mentioned, the extra hydrogen bond that is elicited by oxidizole here imparts a little bit of additional binding to the receptor that we are really happy about. So we use the docking poses as initial poses for the molecular dynamic studies, and for using them as initial coordinates in that case. By coordinates, I mean the XYZ orientation in space that we start with and initiate the molecular dynamics. We got our 3 compounds and did docking for them again against EGFR. In this interplay, we used the crystal structure obtained from the protein data bank 1M17, and the molecular docking of these compounds came up with good scores, and we had variable poses, and we studied the highest populated clusters for each ligand. So for ligand 3D, we found that there is a single pose that we retrieved and said that is highest populated. For compound 9, we had 3 poses that were highly populated, and for compound 12A, we had 2 poses that were highly populated. Here are the pictures depicting the docking results. And we, as we can see, they act as competitive inhibitors. I mean, they bind to the ATP binding site, and they were compared to erlotinib, which we tested, which is in yellow, and this is the one that we tested in vivo. Now we are testing it in the docking. So they can bind and occupy the same active site as erlotinib, as you can see in all pictures, and they have hydrogen bond interaction, variable ones, with the various amino acids. Like here, for example, our compound binds to methionine, which is present in what we call a hinge region. It binds also to threonine, which is what we call a gatekeeper, and it binds to glutamate A31 by lipophilic interactions. It's not shown, of course, it doesn't have a depiction here, but in other softwares we can visualize it. And this is at the activation loop. And the poses, as I said, that were obtained from the docking were used for the molecular dynamics to first investigate what we call pose stability. The MD simulation was run for 100 nanoseconds, and each one of those lines as per the color code, represents one pose. So the red line represents 3D. The green one represents compound 9 pose 1, blue 9 pose 2, yellow 9 pose 3, and so on for all the compounds. And we can find, for example, that pose 1 is really stable. The line has a very small RMSD. On the other hand, 3 was, moving up and down in energy. And this means there is a little bit of a motion inside the active site of 3D when it's binding to EGFR. The blue one pose, sim-wise, all poses for compound 9 were stable. The most stable is the green one. For compound 9, I mean 12A, we had two poses that were being tested, the maroon one and the gray one. The maroon seems to be more stable than the gray one. We did what we call hydrogen bond analysis for all the obtained poses, and for compound 3D, we found that there is hydrogen bond with glutamine 767 that was stable for 89% of the lifetime of the hydrogen bond, which is during the 100 nanosecond simulation. And there was another that was stable for 31% of the time of the simulation. For pose 1 of compound 9, we had two hydrogen bonds that were stable for 31 and 20% of the lifetime of the hydrogen bonds. However, pose 2 showed higher stability, a higher number of hydrogen bonds that were stable for the time of the MD simulation, 56%, 30%, 32%. Pose 3 of 9 had only 2 bonds. So we proposed that the most probable pose for the binding of compound 9 is pose 2. For compound 12A, pose 1 had one hydrogen bond that's stable, but for a short time of the simulation, and pose 2, the hydrogen bonds were forming and breaking. None of them was persistent for long enough. Of course, if we would have X-ray crystal structure of our compounds with the either of the tested proteins, EGFR or COX-2, that would have given us a real story. But since we don't have this facility at our lab, then molecular docking and molecular dynamics, they try to answer part of the question of how strong do these compounds bind to the active site. Last thing that we did, we got our 3 compounds and tested their pharmacokinetic activity in silico using a SUSADME tool. It's a free web tool. Just you need to feed the structure to, and it makes all the calculation based on an algorithm that predicts drug likeness properties and make other calculations regarding molecular weight, the log P, number of nitrogens, oxygens, number of hydrogen bond donors, Lipinski's rule of 5 violations, total polar surface area or topological polar surface area, GI absorption prediction. So we found that all compounds, they have possibility to be absorbed from the GIT, blood-brain barrier permeation. So it was found that the first 2 compounds, they do not penetrate the blood-brain barrier, which is good in our case because we are testing our compounds against hepatocellular carcinoma. Compound 12A would be able to penetrate blood-brain barrier. So if it is to be tested against brain type of cancer, that would be good. Otherwise, that would be a side effect. Pains alerts, all the compounds had 0 alerts. Brinks alert, compound 9 had 0 alerts. 12 had an alert because of the amine, and 3D had 2 alerts because of the amine and the nitro group. The bioavailability score of all the compounds, they are high, not bad. They have good bioavailability, and the lead likeness, they are all lead-like compounds, which means if we want to make further improvement to push them to enhance the pharmacokinetic and pharmacodynamic properties, there is a lot of room for that. So in conclusion, the best compound from this study is compound 9 that has a urea linker and it has a 3-pyridyl moiety at the 5 ends and a phenyl urea at the 2 end. It has a very nice COX-1, COX-2 inhibitory activity with high selectivity towards COX-2. It is a potent cytotoxic agent for UO31. It has potent inhibition effect on EGFR. So we came up with this compound as a promising lead for this series of compounds. So this concludes my first study. I don't know if I have time to go through another study, or should we reschedule it for another time, or whatever you prefer.
Okay, thank you very much, ma'am, for presenting such a nice discovery. Actually, that is a dual targeting strategy that you used in cancer. There, that is very, very impressive. And we also using such type of strategy in our lab because we are also performing 3D QSAR, virtual screening, pharmacophore modeling, and docking and molecular dynamic study. So we can better understand that your drug discovery that you go for the production of that anti-cancer drug. So, ma'am, which software you have used for this drug discovery process?
Would you say that again? The question, the software
Which,
that we used
yeah, which software you used?
for the docking studies, we used MOE, Molecular Operating Environments, version 2019.
Okay,
This one we have, and we had the license for. And for the molecular dynamics, we used Gromix.
Gromix. Okay,
Yes.
okay.
And for some of the visualizations, we use